Start the motor, bring the action, and let's become Michael Jackson. So if you want to cook a punch, or if you want to dance like Allu Arjun, then it must be function of our muscles. Okay, so here meet our haddi, which is inside our body. So in this class we are going to talk about contraction of muscle. So before that, let me introduce myself. I am Biswajit Ghosal, offline zoology educator at Vijayawada Unacademy Center. So here our body cannot dance if there is only skeleton, if there is no muscle. So in order to perform the movement, it needs muscular system upon the skeleton. It needs muscle upon the skeleton, and those muscles are called skeletal muscles, which are kind of voluntary muscles. In order to move those muscles, we need to think from our brain, that is from our will and wish. Before studying the muscle contraction, we need to know about the basic compositions of skeletal muscle. So let's see. Okay, so skeletal muscle is made up of basically motu and patlu. Okay, now we'll say, what is this, man? Motu means a particular protein or thick protein, that is myosin, and patlu means thin filament or thin protein, that is actin. So basically our skeletal muscles are made up of two basic proteins, actin and myosin. Now how this actin is made up of? This actin is made up of numerous globular monomers called G-actin, or globular actin. Now when they combine together like a beads on a ring. Now if you keep combining this G-actin one by one and you make a filamentous chain, that is called F-actin or filamentous actin. Okay, now two filamentous actin wrap each other like snakes. Two filamentous actin wrap each other like snakes and form actin filament. Okay, so this is ultimately the polymer, which is made up of monomer globular actin. Now if you see the actin, what we can see? This actin filament is wrapped up by further two fibers, further two filaments. Now these proteinaceous filaments are known as tropomyosin. Now this filamentous actin helix is wrapped by another filament called tropomyosin. How many tropomyosins are there? Two tropomyosins are wrapping one filamentous actin, means that double helix of one filamentous actin. Okay, now what is the function of this tropomyosin? The tropomyosin actually is blocking the active sites of the actin. Suppose this is the actin filament, actin protein. Now upon this actin protein, there are certain places which we call active sites. Now what are these active sites? These active sites are actually binding site for myosin. Okay, now in normal condition, in resting phase, these active sites gets blocked by this tropomyosin. Now this tropomyosin single individually cannot do the masking, cannot do the blocking. It needs help of another trimer protein. So here you can see there is these trimers. These are called troponin. So this troponin trimer, okay, is helping tropomyosin to bind upon the active sites of actin. Okay, now why I am saying this troponin is a trimer? Because this troponin having three globular protein, right? Troponin T, troponin I, troponin C. Troponin T means this is the segment by which troponin gets binded with tropomyosin. Okay. Troponin I, this is the segment by which it is inhibiting or blocking the attachment of myosin. Troponin C is having affinity towards calcium ion. Troponin C is having affinity towards calcium ions. Okay, so this is the configuration of actin. In normal condition, the actin get inactivated, in terms it get masked by dual function of troponin and tropomyosin. Okay, now what is myosin? Myosin is a thick protein or thick filament, right? The monomer of myosin is called meromyosin. Monomer of actin was globular actin. Monomer of myosin, we call them meromyosin. Now few hundreds of meromyosin come together to build a myosin filament, right? Okay, now let's check the structure of meromyosin. So this is the structure of meromyosin. This is the structure of meromyosin. You can see like snake, two meromyosins wrapping up each other. Okay, now if you see the structure, there is a tail segment, which is also called LMM or light meromyosin. There is a head segment, or you can say globular head segment, which is called HMM or heavy meromyosin. Head is heavier than tail. Okay, now these double meromyosins are crossing each other while making the head region. So this is called cross arm. Do not confuse with cross breach formation during muscular movement. Okay, now we need to study about the head. Okay, the head is itself, okay, it's a enzyme called ATPase. It's having ATP breaking capability in order to produce energy. Okay, so globular head is basically ATPase, acting as ATPase, and it is having two sites, ATP binding site. When ATP will bind with it, sorry, not this is not ATP binding site, this is ATP binding site. When ATP will bind with it, then only it will hydrolyze the ATP and it will break down the ATP into adenosine diphosphate and inorganic phosphate, right? And it will produce energy. So it's having ATP binding site, and these are basically actin binding site, by which, suppose this is the myosin head, this is the actin binding site, this is the actin. Okay, so it will bind with the active site of acting with actin binding site on myosin head, like this manner. Okay, so how this muscle contraction occurs? When you show your dole shole, right? When you show your dole shole, then your muscle contraction takes place into your biceps. So we are going to study in a brief manner how this muscle contraction is going on and what are the mechanochemical procedure responsible for it. Okay, so let's check it. Okay, so now we'll know the meaning of the beginning of this class. How I began: start the motor and bring the action. Let's become Michael Jackson. Okay, start the motor means if this is a muscle cell, so with every muscle cell there is a junction, neuromuscular junction or motor end plate of motor neurons. Okay, motor end plate of motor neurons. Now motor neuron is carrying motor signal towards the muscle, towards the skeletal muscle cells. Now the junction between motor neuron and skeletal muscle is called motor end plate or neuromuscular junction. Now it will transmit the signal from neuron to muscle for to begin the contraction process, to give the signal of contraction process. Now who will be carrying the signal of contraction process? There is a neurotransmitter called acetylcholine or ACH. Now when the motor signal will come to the motor end plate, it will release ACH and it will carry forward the signal inside the sarcolemma, means the membrane of the muscle cell. Membrane of muscle cell, sarcolemma, right? So here the meaning of start the motor and bring the action. So when their motor signal will arrive through motor end plate, it will be releasing acetylcholine from the presynaptic knob, okay, and after that, after the release of acetylcholine, there will be depolarization of sarcolemma. Now what do you mean by depolarization? In normal condition, what happens? Inside there is negative environment, outside there is positive environment. Now when you get motivation, when you get motivated, what happens? Negativity, you eliminate all the negativity from your mind and you are filled and you are recharged with, you are filled with joy. That is called positivity. So the inside environment will become positive and outside environment of the cell will become negative. This is called depolarization. Now when that depolarization takes place, so one part of the cell is depolarized, another part of the cell is still in resting phase or polarized phase, right? Now if this portion is depolarized and this portion is in resting, here positively charged, this portion is positively charged, this portion is negatively charged. Now from here to there will be formation of a local circuit. There will be formation of a local circuit and electrochemical signal will start moving from this zone to this zone. So this is the action, this is a potential, this is called action potential, okay. This is a potential difference which is in action, which is flowing, okay, due to the formation of local circuit inside the cell membrane, okay. Now due to the action potential, what will happen? There are sarcoplasmic reticulum inside the cell. Sarcoplasmic reticulum is a kind of endoplasmic reticulum. They will release calcium. Okay. Now after the release of calcium, this calcium will call the troponin. The troponin is having high affinity towards calcium. ट्रोपोनिन को कैल्शियम लालच देते हैं, ठीक है. अब ये लालची ट्रोपोनिन ट्रोपोमायोसिन को लेके कैल्शियम की ओर भागेगा. एंड व्हाट विल हैपन? दिस विल कॉज अनमस्किंग ऑफ मायोसिन बाइंडिंग साइट. मायोसिन बाइंडिंग साइट के ऊपर वो बैठा हुआ था, अब जैसे कैल्शियम मिला, कैल्शियम की ओर भागा और अनमस्क कर दिया. किसको? एक्टिन को अनमस्क कर दिया. ओके. नाउ as soon as there is unmasking of actin, actin is free to interact with myosin. Myosin will catch ATP, it will hydrolyze ATP, it will break down ATP and it will produce energy. And by that energy, when it get recharged with the energy, it will form a cross bridge with actin. It will form a cross bridge. It will attach. It will get attached with the actin. That is called cross bridge formation. And ultimately it will pull the actin filament towards the M line, towards the middle zone. Okay. So this is basically a rough flow chart of sliding filament theory, where it is explained how muscle get contracted. Now here we will see how the things are going on. So this is actin. Okay, so this is actin and this is myosin. Now due to the influence of this calcium ion, now the binding site, the active site of actins are freed now. Okay, because the tropomyosin with troponin slided past. Okay, so as soon as it become free, the active sites of the actin, this is the myosin head, it will catch ATP, it will break down the ATP. It will catch ATP and it will break down into ADP and PI or inorganic phosphate, and there will be formation of energy. Now this will utilize the energy and it will bind, the head will bind to the active site of actin, and this is called cross bridge formation. So it initiated with unmasking of actin. पहले actin unmask हुआ, after that there will be formation of cross bridge. Now as soon as there will be cross bridge formation, it has to pull the actin filament, towards the center. Okay, so it's pulling the actin filament into this direction. Okay, so this is called sliding, or power stroke. This is called sliding or power stroke. So if you study the sarcomere, the functional unit of muscle, fiber, so these are Z lines, these are actin, this is myosin. Okay, so myosin will catch the actin filament and pull towards the M line, So Z lines or Z disc will come closer to each other. There may be obliteration of I band, but there won't be any effect in A band. Okay, you need to remember this. And you need to remember one more thing: during the time of muscle contraction, there is no change in the length of actin as well as myosin. Okay, I am repeating again, during the time of muscle contraction, there is no change in the length of actin and myosin. मोटू पतलू के लेंथ में कोई चेंज नहीं होगा, बस एक दूसरे को पकड़ के खींचेगा. ओके, अब एनसीईआरटी का एक लाइन याद रखना: पतलू ऊपर मोटू नीचे, मोटू पतलू को अंदर खींचे. पतलू ऊपर मोटू नीचे, मोटू पतलू को अंदर खींचे. तो there is a question that actin slides over myosin and myosin pulls the actin. तो यहाँ पे sliding हो गया, या power stroke is completed now. Now after this power stroke, myosin head will lose its energy. Okay, it will be releasing the ADP and PI, so there will be breaking of cross bridge. There will be detach. Head of myosin will get detached with actin active site, and hence there will be relaxation. Okay, and again it will bind another ATP. It will hydrolyze the ATP, and the process will go on. As soon as until unless you are empty in your ATP storage, you are empty in your calcium storage. जब तक एनर्जी है तब तक मसल कॉन्ट्रैक्शन होते रहेगा, एंड व्हेन एनर्जी गेट्स एंडेड एंड व्हेन देयर इज जो मोटर सिग्नल आ रहा था, देयर इज हिंड्रेंस इन मोटर सिग्नल, और व्हेन मोटर सिग्नल transmission gets stopped, then the process will also get stopped, right? Okay, now so this is called muscle contraction, because of this all the processes, Z disc will come towards each other, and that called shortening the length of I band and shortening the length of sarcomere, but not length of actin as well as myosin. Okay, now when calcium ions will pump back into the sarcoplasmic reticulum, When calcium is absent into the sarcoplasm, what will happen? Again this troponin trimer will get addicted to the active side of this actin filament, and it will bind on that particular side. Okay, and it will cause again masking, and here there will be relaxation of muscle. So this is the end of today's lecture, and thank you everyone.